Hi, my name is Jose, and today we're going to talk about the citric acid cycle. That's right, the dreaded citric acid cycle. The one that you see in textbooks and everywhere else on the internet with a bunch of names and arrows and terms and a lot of jargon that it's so difficult to memorize. But I'm going to show you a very easy way to remember the intermediates of the citric acid cycle, the names of those enzymes, and what the whole thing does as a whole. So let's get started. The citric acid cycle is most commonly known as the Krebs cycle, as most of you know, but less commonly known as the tricarboxylic acid cycle, or the TCA cycle. And this takes place in the inner mitochondrial matrix of the mitochondrion. This process, in total, involves a series of successive reactions that start with citric acid, or citrate, hence the name, the citric acid cycle. Its main purpose is actually to degrade the 2-carbon unit of acetyl coenzyme A, or acetyl-CoA, into 2 molecules of carbon dioxide. But it specifically contributes to aerobic respiration in 2 very important ways. The first one is to form your electron carriers by a reduction reaction, which is a type of redox reactions where you add electrons to a compound. In this case, we have NAD plus obtaining 2 electrons to form NADH plus H. So these 2 electrons don't just come from nowhere, they come from hydrogens, which are added to the molecule. Then you have FAD being reduced, again with 2 electrons, to form FADH2. The second way that this process contributes to the cell is by synthesizing, of course, ATP, which is a very high energy molecule, and it does so through a process called substrate-level phosphorylation. So, let's get to the meat and potatoes of this process. So we're going to play a little game called Follow the Carbons, 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 as you've heard in another song of a similar nature. We start off with a 6-carbon molecule, which we know as glucose, right? So let's draw our 6 carbons. That's cleaved into 3 carbons, or pyruvate. And the carbon count is further reduced when you convert pyruvate into acetyl-CoA. So now our carbon count is 2. So why did I draw 2 cycles? Because for one glucose molecule, this cycle occurs twice. And it generates 4 major molecules that are needed for later processes. Our first one is NAD+, and 3 of those are converted into 3 NADHs. 1 FAD is reduced to 1 FADH2. 3 water molecules are then used to convert the respective intermediate into 2 carbon dioxide molecules, which again came from our acetyl group. And finally, 1 ADP is phosphorylated to form 1 ATP. Now this same thing happens in this cycle. So we can total that up to make 6 NADHs, 2 FADH2s, 4 CO2 molecules, and 2 ATPs. And this is all per 1 glucose. Simple enough, right? But that's not the toughest part. Now we're going to get to the part where we have to name the enzymes, our products, and how many carbons those products have. We start off by considering the fact that each product's name is the name of the enzyme that comes after that. So what I mean is, here we have isocitrate, and the enzyme that uses isocitrate happens to be called, very conveniently, isocitrate dehydrogenase. The enzyme that uses alpha-ketoglutarate is called alpha-ketoglutarate. And this pattern carries on all the way until malate dehydrogenase, luckily for you. Malate dehydrogenase makes oxaloacetate. Oxaloacetate is an important regenerating molecule because that last intermediate is what's used in conjunction with acetyl-CoA to form citric acid. Now, it's important to note the number of carbons as each of these reactions take place. So the first two intermediates are both going to start off with 6 carbons. It's going to lose 1 carbon, isocitrate's going to lose 1 carbon in the form of, can you guess what that is? That's right, carbon dioxide. So the first carbon dioxide, which came from acetyl group, is going to be lost as CO2 here. This molecule is then going to lose another carbon, again in the form of CO2, and that's where the second CO2 molecule comes from. And finally, this number of carbons remains the same for the rest of the intermediates. So the rest of these molecules will all have 4 carbons. Now, these are all very complicated names, and it's important to preserve the order when you're trying to memorize them. So I've created some, what I believe are funny mnemonic devices to help you remember the names of each of them in order. So for our Spanish-speaking students here, I created one here called Sidra Aconita is at Suki Suki Bar, fumando y maltratando. So, I hope that got a laugh, but if it didn't, that's okay. I hope it's at least memorable. The first few letters of each of these words happens to be the first few letters of each of the enzymes, again, in order. So for our Spanish-speaking individuals, I have an English version here that goes, Citra Agonita is at Suki Suki Bar finding men. And why not? It's a Friday, right? For our products, I have another neat mnemonic device that helped me when I was actually taking an exam on this. That goes, can I keep selling substances for money, officer? Again, I don't encourage you to speak to law enforcement officers like that, but it works if you're taking an exam.